So we are going to cover competency 10.1. This is, you can see, about the cancer. And today we are going to talk about, oncogene, anti-oncogenes, etc. So we are going to focus on oncogenes, the cancer-producing genes. These are generated by activation of proto-oncogene. This is actually oncogenes are the genes, the protein product of which causes cancer. It is growth promoting and it causes cancer. So proto-oncogene to oncogene, this is called oncogenic activation because it is cancer producing mutation. So the topic for today's discussion is activation, mutational activation. Of course, we are going to discuss of proto-oncogene to oncogene. Now, this word pro, this is reminiscent of the word precursor. We entertain a slight digression to understand this concept. You have already done about pro-insulin, the precursor form comprising of long A chain, B chain, and a C peptide. Removal of this C peptide results in formation of insulin, 20 amino acid, 30 amino acid long 2 peptide chain joined by disulfide bridges, of course. But the important point here is that the pro form is converted to the final insulin and the mechanism is proteolytic activation. Likewise, pro-parathormone, PTH abbreviated, converted to fully active parathormone by the same mechanism of proteolytic cleavage, removal of a peptide group. What about proto-oncogene to oncogene activation? Is it also proteolytic cleavage or it is something else? No, it is not proteolytic cleavage. It is something else. What we are going to discuss today, how proto-oncogenes get activated to oncogene. We must outline some characteristics of proto-oncogenes. So this slide outlines everything. These are normal cellular genes and they code for growth promoting proteins. And these proteins are pro-mitotic also. And of course, other functions are also there. But at the moment, for today's lecture, pro-mitotic and growth promoting, these two words are important. And one more point, that proto-oncogenes are under meticulous control. They are under regulation. So they are expressed only when they are required. This point needs elaboration. I'm going to talk. You can take down if you want. They are linked in the signaling pathways. They serve as links in the signaling pathway normally. So, अब ये mitosis. All of you know it very well that normally the life starts from a single cell called zygote. There are multiple divisions and then large number of cells are formed and they get differentiation. They get differentiated into various forms, cuboidal form, squamous form, etc., etc. So division and differentiation and everything, as I said, is under meticulous control. Now, for example, a single mitotic division, what we had shown, it comprises of 4 major phases. And these phases are based on biochemical changes, not on the cytological changes. One, this is the replication phase. M, this is the mitotic phase. And then there are certain gap phases also. And some cells like nerve and muscle, they go into G0 phase. They do not divide at all. But other cells, for example, hepatocyte, fibroblast, etc., they undergo this repeated cell division. So at the moment, we are heading towards our aim to understand what are proto-oncogenes and what is their role in the how they are serving as the pro-mitotic protein. So in this rough figure, I have tried to show you that there are, as I said, everything is under meticulous control. Meticulous control of what? Of certain chemicals which are present in very minute quantities. We call them mitogens. Mitogens, for example, growth factor, exemplifying here. Growth factor is not a single protein, it is a group of more than 100 proteins which interact with the respective receptor. And then by a series of changes, the cascading pathway, ultimately they converge onto cyclin and CDK proteins. These are the proteins CDK, cyclin dependent kinases, which cause phosphorylation of multiple intracellular protein and which help in progression of the cell cycle. तो अभी तक हमने बात की that cell cycle progression or the mitotic division is under control of the growth factor. And it is not a simple event. It is a cascade of events, cascade of multiple steps where various proteins they participate. For example, a G protein called RAS, REF, MIC, a number of others are also there, but that is not our topic today. So all these are called proto-oncogene. Even I would say that the growth factor receptor itself is a proto-oncogene, or you can call growth factor also proto-oncogene. So REF, for example, so REF, you can see it is an important link in the signaling pathway leading to progression of the cell cycle, leading to the mitotic division. So we will call REF as proto-oncogene. This RAS make a number of others also. So these are all proto-oncogenes. So I think now, so these are normal cellular genes, and what are they? Links in the mitogenic signaling pathway. And they are under regulation. And we saw that they code for pro-mitotic proteins. Pro-mitotic proteins, a number of phosphorylation events, etc. We are not going to go much into that now. For the time being, proto-oncogene ka activation to oncogene, that is the cause of cancer. So oncogene is abnormal form of proto-oncogene, protein product of which causes cancer. So ये जैसे for example, REF, REF is a proto-oncogene. It is possible that the REF undergoes point mutation. Point mutation के बाद क्या बन जाएगा? A super active form of REF is formed. A super active form. This super active form is active all the time. देखिए, please note this point. REF for its activation required stimulation by the mitogen growth factor. But this activated REF, this does not require any stimulation. ये persistent active होता है. So this is an oncogene. Okay. So persistent activation, not under control. A very big example, it is normally said that oncogenes are like rabid dogs. They will bite all the time. Whereas proto-oncogenes, they are chained dogs, paltu. They will, bite. They are totally under control. Okay. They are capable of growth promoting effect, mitosis, but strictly under meticulous control of the mitogens, but not the oncogene. They are totally independent. So proto-oncogene के हमने ये characteristics 4 देखे थे. Now we are able to say what they are. तो mutational activation कैसे होता है? The first one we have already considered, point mutation. Point mutation in the any of the links, RAS, REF वगैरह में रेस का भी होता है. I have told you about this thing, REF. Now let me talk about RAS a bit. RAS actually it is a G protein. So all of you know about this G protein. You have come across the G protein when you were studying hormones. So there are trimeric comprising of three different subunits. But RAS, it is a single subunit. Only one subunit, monomeric protein. और बिल्कुल उसी तरह से G protein था. It is present mostly in the normally in the GDP bound state एंड एक्टिवेट होने के बाद ये जीटीपी बाउंड स्टेट में चला जाता है. तो रेफ के अंदर भी म्यूटेशनल एक्टिवेशन अनकंट्रोल्ड अनहिंडर्ड रेस की एक्टिविटी जीटीपी बाउंड स्टेट वो परसिस्टेंटली प्रेजेंट होती है एंड दैट कॉजेस persistent oncogenic activation etc we talked about. तो पॉइंट म्यूटेशन में हमने दो एग्जांपल देख लिए, रेस और रेफ का एग्जांपल. वन मोर एग्जांपल आई कैन गिव यू. अभी तक हमने देखा जैसे ये रेस है फॉर रेफ, ओ माय गॉड इट्स कंफ्यूजिंग, रेफ को हम कर रहे हैं. तो द म्यूटेशन इज नॉट इन द रेफ जीन, द म्यूटेशन इज इन द प्रमोटर. All of you know, expression of REF is under stimulatory control of the promoter. You know it very well. ऐसा भी हो सकता है that the point mutation is occurring in the promoter. So as a result, this positive influence becomes too much. All right. So in this case, no superactive REF is formed, but REF is overexpressed. All right. तो पॉइंट म्यूटेशन दो तरीके से हो सकता है. आइदर इट प्रोड्यूसेस अ सुपर एक्टिव प्रोडक्ट और द प्रोडक्ट इज ओवर एक्सप्रेस डिपेंडिंग ऑन वेदर द पॉइंट म्यूटेशन इज इन द कोडिंग सीक्वेंस और इन द प्रमोटर सीक्वेंस. नेक्स्ट वी कम टू we are now presently studying the mechanisms of mutational activations. First mechanism हम consider कर चुके हैं. Now we are coming to the second one, gene amplification. So all of you know gene amplification का क्या मतलब है. Multiple copies of a given gene are produced. So normally ये for example one copy is there that produces for example x amount of it. अब कितनी हो गई, 7 हो गई, तो 7x. Multiple copies they produce greatly increased amount of the protein. तो कुछ जीन्स का एम्प्लीफिकेशन हो सकता है. जैसे हमने देखा था कैस्केडिंग पाथवे के अंदर एक इंपॉर्टेंट लिंक इज मिक MYC. Various forms of this MYC are present. I'm right. MYC are present. So in some cancers, amplification of the MYC gene are seen. Particularly, there are three different forms, N-MYC, C-MYC, etc. N-MYC का amplification हो जाता है, and then multiple copies. So this is what overexpression of a growth promoting proto-oncogene. So I am going to draw clinical correlate. There is a Burkitt lymphoma. So to understand this, we will have to understand the third mechanism, gene translocation. Gene translocation का क्या मतलब है? You see, every gene, as we said, ये यहाँ पर हम मिक कर देते हैं. मिक gene is under control of the promoter. Promoter का क्या है? Just can I just take a minute? इसको delete कर दिया. MYC gene is under influence of its promoter. So gene translocation का क्या मतलब है? ये इसका promoter का positive influence है and it is well under meticulous control as we have been saying. But is it possible that the MYC gene totally it is lifted up, broken from its original site and shifted to some alternate site where it is under influence of a very strong promoter? तो इससे क्या होगा? The promoter is very strong. It is providing 10 times, 20 times more positive signals to MYC gene for its expression. So a gene is lifted from its original location and shifted to an alternate hotspot location where it is overexpressed. So this is the third mechanism, gene translocation, and this is what happens in Burkitt lymphoma. Activation of CMAC. All right. So CMAC, it's a transcription factor, a proto-oncogene, of course, and this is an important link in the cell cycle progression. So activation क्यों हुआ? Because of gene translocation to an transcriptionally active site, we called it hotspot. So promoter, for example, of immunoglobulin heavy chain, उसके अगर hot, but that's a very strong promoter. Immunoglobulin kappa chain, lambda chain, they are hot spots. They are very strong promoters. So it is overexpressed at the new site under influence of, I have written not promoter enhancer here. This is promoter enhancer, उनका action same है. Please find out what is the difference between the two. All right. तो अभी तक हमने जो बात की इसके अंदर क्या था? Gene is lifted and translocated to a site where a strong promoter exists. ये भी हो सकता है क्या कि जीन अपनी ओरिजिनल साइट पर ही रहे और प्रमोटर यहां पर आकर इंसर्ट हो जाए. Is it possible? Yes, it is possible. It happens by viruses, virus oncogenesis. As you people know very well, viruses, they insert their genome into the host cell genome. तो वायरस का जीनोम होस्ट सेल के अंदर, जैसे रेट्रोवायरस है फॉर एग्जांपल, रेट्रोवायरस का कॉम्प्लीमेंट्री डीएनए बना, complementary DNA by help of an enzyme integrase is integrated into the host cell genome. तो अब वायरस का प्रमोटर भी इंसर्ट हो गया. तो अब इससे क्या होगा? Now I will show here. This is a cellular proto-oncogene. So the viral genome is inserted in immediate neighborhood of the cellular proto-oncogene. तो ये LTR, ये promoter sequences है वायरस की. So there will be over expression and excessive production of oncogenic product. This is causing cancerous transformation. Okay, मैंने अभी आपसे थोड़ी देर पहले क्वेश्चन पूछा था एनहांसर एंड प्रमोटर का. यहाँ पर एनहांसर है, नॉट प्रमोटर. और फर्क ये है, you concentrate on this slide only. Promoter is present upstream of the proto-oncogene at a particular distance. I think it is 25 nucleotides, Dr. Adel, isn't it? But enhancer has got no limitation. It may be present upstream, downstream, at a great distance, even thousands of nucleotides away, even in the wrong orientation. So enhancer that has got much more flexibility. But हमारा इस समय point ये था, हमने अभी ये किया था कि the another mechanism we have studied. First हमने देखा था point mutation which can cause superactive product, the oncoprotein. And we also saw और एम्प्लीफिकेशन ने क्या किया था? ओवर प्रोड्यूस प्रोडक्ट, ओवर एक्सप्रेस. थर्ड हमने देखा था ट्रांसलोकेशन ऑफ अ स्ट्रॉन्ग एनहांसर और अ प्रमोटर इंसर्शन. तो ये हमने वेरियस मैकेनिज्म वी हैव सीन. द फर्स्ट वन इज कॉजिंग सुपर एक्टिव प्रोडक्ट. Next three are causing overexpressed or overproduced product. But one point I may repeat, the point mutation also can overproduce, result in overproduced product whenever it is in the promoter or the enhancer sequence. Okay. So this slide summarizes everything. And we have taken one clinical example, Burkitt lymphoma. At conclusion, I will take two more examples just to summarize, to bind everything, thread everything into few words only. तो growth promoting oncogenes क्या होते हैं हमने देखा, growth protein promoting proteins को करते हैं produced by mutational activation of certain proto-oncogene, और उसके हमने 4 या 5 मैकेनिज्म हमने देख लिए. All right, anti-oncogenes we will be studying some other time, maybe in the next class. So now we are going to do another form of translocation. So take down, another translocation is involved in formation of Philadelphia chromosome. तो अब हम ये देखने वाले हैं what is Philadelphia chromosome. सर्किट में हमने क्या देखा था, ट्रांसलोकेशन हुआ था, I may completed. Chromosome number 8 was translocated to chromosome number 2 or 22 or 14. जहां पर in this condition chronic myelogenous leukemia, वहां पर मिक जीन हुआ था, यहाँ पर another proto-oncogene ABL1 proto-oncogene. It codes for certain of protein kinases which are participating in the regulation of the cell cycle. तो यहाँ पर क्या होगा, it is overexpressed. ट्रांसलोकेशन क्या हुआ, 9 and 22. मेरे ख्याल से 22 का पोर्शन it is broken and shifted to 9. So as a result, new fusion gene is formed that is called Philadelphia chromosome. It comprises of a small piece of PCR, a small piece of PCR gene and major portion of ABL. So ABL1 is under influence of a PCR, which is a promoter gene. So PCR promoter के ओवर इन्फ्लुएंस में ओवर एक्सप्रेशन हो जाता है. तो यहाँ पर क्या होगा, अ फ्यूजन प्रोटीन इज फॉर्म. तो फ्यूजन प्रोटीन has kinase activity and it is over expressed, and the dysregulated kinase causes abnormal cell proliferation. So it is like Burkitt lymphoma, except the fusion protein is formed. So this mechanism, instead of gene translocation, at some places this is also called gene fusion. एक और condition, neurofibromatosis, with the help of what we have studied so far, I'm going to discuss. अभी हमने देखा था कि जैसे लिंक होता है माइटोजेनिक सिग्नलिंग में, growth factor receptor, and then the first link is a G protein called RAS, and we also know about the G protein. This is RAS. We also know about that in the inactive state, they are in the silent cell, they are bound to GDP. After mitogenic signaling, GDP is replaced by GTP, and this is the activated form. Now, after the work is over, it must, as we said, इसको तभी act करना है जब signal आ रहा है. So the GDP bound form is getting converted to, I'm going to write again, I think, GDP form is converted after mitogenic signaling to the GTP bound form. After it is over, the GTP form must be converted to the inactive GDP bound form. और यहाँ पर कुछ GTP ages they are active. Next slide में थोड़ा more elaborate. You can see here growth factor binding with the receptor, some changes. RAS GDP is converted to RAS GTP, and this is RAS GTP is calling the cellular response of proliferation. तो हमने अभी बात की GTP must be converted to GDP. तो GTP is activity होती है. So GTP is activity is promoted by these two proteins, GAP and neurofibrin. So supposing if there is mutation in the neurofibromin, so then RAS GTP will not be converted to RAS GTP. Reduced conversion or even absent conversion. So क्या होगा? It is locked in the activated GTP bound state. Locked in activated form simply because it has lost its off switch. Off switch khatam ho gaya, so it is locked in the activated GTP bound state. So persistent activation will be there. And this is cause of a disease which we call neurofibromatosis. Enhanced signaling through RAS because the RAS is locked in the activated GTP bound state. Loss of the off switch. ये off switch का loss हो गया. All right. So I think all these points we have exemplified with 3 clinical examples also. So this is to show you in a more elaborate manner. हमने ये 3 को आज डिस्कस किया, RAS, REF and MIC. A number of others also are there, so which you will come across, the inquisitive students. You can read them. I would recommend certain textbooks by Rafi, Dr. S.K. Gupta, or if you are lucky, you can refer to uh, some other book also. All right. Harper, of course, is the uh, proverbial Bible. So, cyclin CDK पर converge हुआ. तो लास्टली हम ये एक और बात करने वाले हैं, सम एडवांस पॉइंट फॉर द इंक्विजिटिव माइंड्स. हमने देखा था जीनोम, रेट्रोवायरल जीनोम इंटेग्रेट हो गया. Sometimes it happens that the retrovirus contains a cancer producing gene. Retrovirus contains a cancer producing gene. So this is an enigma. This is mysterious. How a viral gene can cause cancer in human. So kuch virus, raw sarcoma virus, it contains a gene called SRC, SRC. The viral form of it is called VSRC. And SRC is an important proto-oncogene in humans also, and this is called CSRC. So ये थोड़ा mystery थी which was solved actually, how a human proto-oncogene went into virus. So it is now known that during a previous infection cycle, the virus, it picked up a human gene. So the human gene was hijacked by the retrovirus and it was slightly mutated. And that is cancer causing. A proof for this is that VSRC does not perform any function in humans. So it is a hijacked gene in virus. It does not perform any function. In humans, of course, it is deleterious function of cancer. So कोई function नहीं virus में. So it is a hijacked gene, hijacked during a previous infectious cycle. So this is from where we started. The last thing, there are 2 ways cancer can be caused, either by oncogenic activation of proto-oncogene or by inactivation of a anti-oncogene. Anti-oncogene which depresses, which has got a negative influence on the cell cycle progression. It is like brakes and accelerator. आपको अपनी कार तेज करनी है. Either you press the accelerator or you apply the brake. Anti-oncogenes are like brakes. P53, retinoblastoma, these two are anti-oncogene. इनके inactivation से भी the same result can be there, cancerous transformation. Last of all, thank you so much.